right. Welcome, Dr. Pack. Thank you so much for coming today.
Okay.
If you could just share us your name, the university, college you teach, and also give us an explanation of your field.
Okay. So my name is Allan Pack. I'm a professor at the University of Pennsylvania in the Division of Sleep Medicine. My research is focused on sleep and sleep disorders. I study genetics of sleep, why we sleep, and then in the more translational stuff, I study obstructive sleep apnea, and it's a very common sleep disorder, and we're trying to develop a more personalized approach to that based on genetics and biomarkers.
Great. So how long have you been a professor?
I've been in the faculty at University of Pennsylvania since 1976. I started as an assistant professor, then became an associate. And I don't recall how long I've been a full professor for, but it's quite a while.
I see you're very dedicated. That's
Yeah.
nice. So do you have any outside interests or hobbies?
Oh, yeah, my family's a big interest of mine. I have four kids and 11 grandchildren, so that's a big interest of mine. I do play golf. I'm not very good at it, but I try.
Yeah, I see. That's yeah, that's great.
Okay.
All right. So what led you into your field of study, and where do you think it's headed towards in the future?
Yeah, well, no, that's a great question. So I mean, I came to United. I grew up in Scotland. I went to University of Glasgow, where I went to medical school and got my PhD. I came to University of Pennsylvania in 1976. I didn't study sleep at that time. In the early 1980s, sleep medicine just got going. I basically, we did the first sleep studies at University of Pennsylvania in the 1980s, got a sleep center going, and really found it a very fascinating, really interesting area. And over the years, it just got better and better because we got more and more tools to study sleep. I think where we're going at the moment, as I said, is trying to really change completely the way people approach this common sleep disorder, obstructive sleep apnea, trying to do that. And we're trying to understand one of the fundamental questions about sleep is, why do you sleep? Why do you spend a third of your life doing this? And we're sort of interested in that. And we're very interested in identifying genes that regulate sleep. There's a lot of variation in sleep patterns in humans, a lot of that's genetic, and we're trying to understand that.
Great. So could you give us a gist of your educational pathway?
What do you mean?
What you majored in and
Oh, right. Yeah. Well, no. I mean, it's Britain, Europe in general is very different than the United States in the way it works, because you go straight from high school to medical school. You don't go to college.
Oh,
So you go straight from high school to medical school, and then medical school is 6 years. The first 2 years are basic science, and then you get into the more clinical stuff. So I went to medical school, I think, when I was 17. And I was 23, I was a physician. and then I was working, and then I did a PhD part-time. I did a PhD in mathematical modeling. I learned a lot of maths and stuff like that. Control engineering is what I did. And then it was hard to, the research opportunities in Britain, particularly in Scotland, were quite limited, and I got offered a job to come to the University of Pennsylvania. I took the job and I moved here in 1976.
That's great. Wonderful. I didn't know about this system that happened in Europe. Oh, that's great.
Yeah, it's completely different. I mean, a lot of the world's like that. America has this thing where you go to college and then you go to medical school. And typically what happens is you go to college, you do 4 years in college, then you take a couple of years out, you're tired of being in classes, and then you go to medical school, then you become a resident and intern. By the time you get going, you're 35 years of age.
Exactly. Yeah, it's so long.
It's too long. It's too long. And I think college is great, but I think you have to think about it. I mean, I don't believe I missed anything by not going to college. I take a keen interest in history. I read lots of politics. I read lots of history. I keep up on all of that. And so I don't believe I lost out by not going to college. I got it in other ways. I mean, part of college is obviously the whole socializing. You get to you leave home, you get to meet people, and you mature in a sense. You know, and that was it was hard when we were in medical school. I mean, we were just kids running around partying and going to medical school. It was quite something.
I see. Great. All right. So my next question is sort of like a broad question, but you can go deep into this if you like. So my question to you is, what questions are you answering in your research if you did anything in the past or for the future? And also any data or latest findings in your lab?
Yeah, no, I think these are great questions. So basically, we continue. I mean, the thing about science is you've got to continue to reinvent yourself. You can't just hope that whatever you know now is going to last for the next 20 years. You have to continue to reinvent yourself. And I've done that over the years multiple times, right? You know, when I came to the United States, I'd never done any animal experiments in my life. I learned how to do that when I came here. I did maybe 15 years ago, I did a course at Jackson Labs in Maine on genetic analysis. I learned a lot of genetics, mouse stuff. And then, you know, one of the things we're proudest of at Penn is we took the view that's, you know, up until the year 2000, all sleep studies were done on mammalian species, typically rats and cats. And people didn't think that any other species slept. And we took a much more aggressive view of that, if that's the right word. We said, you know, there's behavioral, you may not need the EEG, the wires in the head, and looking at brain waves to decide whether you're asleep, and you can develop behavioral criteria, which we did. And we decided in 2000, we were the first group to describe a sleep state in the fruit fly Drosophila, which gives you enormous power because there's so many genetic tools. Then probably 2005, we described sleep in the worm and C. elegans, the worm. Other people decide it in zebrafish. So now we've got all the different model systems, and you can move really fast. And then new approaches have been developed. You know, for example, we just recently described a new gene regulating sleep, and we think we found a new chemical that regulates sleep based on that gene. And that was based on what's called diversity outbred mice. So my colleague at Jackson Labs, he did something which is really revolutionary. So basically the way people use mice was you had all these inbred strains, they were all the same genetically, but they were all different to each other, and you compared them and you tried to find out what genes were in one strain and not the other strain and so on. And what he did random breeding. So he had 8 founders and he did random breeding for multiple generations. So each mouse that goes out at the end is unique, right? You're not going to replicate it. But all the DNA comes, it's all chopped up, but it all comes in some way from these 8 founders. And then you can screen these mice, look and then find out if there's, like what we did, is found there was a piece of the DNA that related to sleep. And then you could drill down in that and you come up with causal genes. So we're very into using these new genetic approaches, diversity outbred mice. We just recently established a zebrafish lab because you can change gene expression very rapidly. Within a week, you can get new fish with a gene knocked out, and you can screen them. And you have a camera, and so you put these fish in a 96 well plate, and you have one camera, and it's following each mouse, each fish rather, and we can study. We have 4 of these boxes, so that means we can study over 300 fish simultaneously, and we can study sleep. And then we can compare the ones that we're knocking a gene out, the ones we don't, and you can move very fast. So the technology in that end is amazing. In the human end, what a lot's happened is people have used these big biobanks. People like Penn and Geisinger and Vanderbilt and so on, they built these large biobanks where you get DNA from people. So people they go to visit their clinic, they give blood for a DNA, and then people do all the genetics. They do a whole genome sequencing, you know, all the genes. And then what you do, and we've just completing a study where we looked at the genetics of sleep apnea. We never recruited any of these people. We found them in EHRs. So we went into the electronic health record and said, who's got sleep apnea? We developed an algorithm to identify in a very, very, validated way. And then we could then pull in the genetics. So we could do genetics. In that genetic study, there's over 20,000 people with sleep apnea and genetics. and we never recruited one of them. So you can move really quickly. So in the human end, what's driving a lot of it now is use of electronic health records for data and biobanks.
I see. So are there any harsh chemicals in the brain that are produced if we don't get enough sleep?
Yeah, well, I think the question is, the question is what happens if you don't get enough sleep, right? So what we believe, and I think most people now believe, is that sleep has got, some people think it's got one essential function. I believe it's got multiple essential functions, okay? And let me go over some of them. So one of the things that happens in sleep is you consolidate memories, right? Interestingly, if you're trying to solve a puzzle and you're not quite there, you go to sleep, you wake up in the morning and you do it better, right? Because your brain's been processing this and you consolidate memories across the night. So that's one thing you do. At a biochemical level, what happens is that during the day, when we're wandering around here, we're producing what's called free radicals, because you're firing off to make energy, ATP, to provide the cells with energy. And part of that, when that electron transport train gets going, it throws off free radicals, which are damaging. I mean, they bind to chemicals and modified proteins and lipids and so on. So when you go to sleep, what happens is the antioxidant enzymes go up and you clean up all these free radicals. So you're building free radicals during the day, which is not good. You go to sleep, you clear it up. The other thing that happens is that when you produce free radicals, it leads to breaks in DNA. So you get damage to DNA. And then what you find is the DNA repair mechanisms go up during sleep, right? So you're damaging your DNA when you're awake and you're mopping it up during sleep. So there's really specific things going on. And then if you stay awake for a long time, every animal is programmed to stay awake for a certain amount of time. Mice don't stay awake for long, an hour at a time, a couple of hours at the most. Humans, we stay awake for 16 hours. Once we get beyond 16 hours, we start getting impaired. You get to 20 hours, you're behaving like somebody who's at the junk level. And so our brains are programmed to be able to cope with 16 hours of continuous wakefulness. And then beyond that, you get into trouble. Now, if you keep mice awake, you keep them awake by, interfering with them, stroking them with a brush or something, keep them awake for a while. What you find is you get a phenomenon called ER stress. So basically proteins are folded in the endoplasmic reticulum. You make them and then they're conformed in there and then they're sent out to where they're supposed to go. Now, if the proteins don't get properly folded, you get what's called the ER stress response or the unfolded protein response. Molecules are upregulated, they come in, they try to get the proteins folded. If they can't get them folded, they take them out for destruction and into the cytoplasm for destruction. And you find that all of these molecules in the unfolded protein response are upregulated with, say, 6 hours of sleep deprivation in mice. And that's been shown in other species as well. So you lose certain essential functions if you don't sleep. But in addition to that, there's this other process that kicks in, the ER stress response. So sleep loss is not innocuous. It really, at a molecular level, is really causing problems.
So I heard that the sleep, if we have sleep loss, it not only affects the brain, but affects the gut. Is that correct?
Yeah, it affects other organs. Yeah, if you go out there and we did a study where we looked at heart and lung, and then there is, as you say, indications in GI system. Our study, we looked at changes in gene expression between sleep and wake and keeping mice awake, sleep deprivation. And we found the ER stress response stuff went up peripherally as well. So there must be some signal coming out from the brain. We don't know what it is telling the system to do that. Yeah, so there's no doubt. I mean, there is a classic paper in our world, which I think is wrong. The guy just died last year, Hobson, and the paper was called "Sleep is by the brain for the brain." So it's very much a brain-centric phenomenon. That's inaccurate. Things out there in the periphery change in every system with sleep.
So how are like brain waves, like theta and beta, processed in the brain, like during the REM sleep cycle? I know we go through like stages of sleep cycle, right?
Yeah. You have these different stages of sleep. You have what's called non-rapid eye movement sleep, and then you have rapid eye movement sleep. Non-rapid eye movement sleep itself is divided into different stages. Used to be N1 to N4, now it's N1 to N3. They collapse 3 and 4 into 1. And what happens, for example, you get a phenomenon of these spindles. You see them in the EEG, sleep spindles in stage 2. And then when you get into the deeper stages of non-REM, you get these big slow waves. So the cells are all synchronized and they're all fired, they're oscillating, right? They're cycling, but they're all cycling together. That's why you get these big waves in your EEG, and that's non-REM sleep. REM sleep is a stage where it's an odd stage of our lives. It occurs in the way the human, mice, the cycles are very rapid. The human cycle in about 90-minute cycles. So you start awake, you go into stage 1 non-REM, stage 2, stage 3, and then REM, and then back into the non-REM. And the REM episodes occur every 90 minutes or so. As the night goes on, the REM episodes get longer. So non-REM is front-loaded to the beginning of the night, and REM is back-loaded towards the end of the night. Now, what happens in rapid eye movement sleep is you're paralyzed. So you're completely atonic. The brain sends signals out and shuts the muscles down, so you don't move. So you're atonic. Your brain is very active. There's lots of things going on, flurries of activity. That's the stage you dream. If you wake people up, they're dreaming. So believe you dream in REM sleep, and you see these flurries of activity. And the reason it's called rapid eye movement is because you get these twitches. If you see a dog in REM, you'll see his paw twitching. Here, your eyes are flashing, right? So you're getting this phasic stuff coming out from the brain, and it's strong enough to break through the atonia, the paralysis. And you also get enormous variability in heart rate. So heart rate is very variable, breathing is very variable. So REM is a very interesting stage of our lives. But basically, you're paralyzed and the brain's very active. Recently, there's a paper just come out last year in Nature by a guy, Moraine, in Stanford, where he's indicated he's found a REM sleep in zebrafish, right? I mean, the mammals and birds were describing REM sleep, never been described in these other systems before, but he did a very cool thing, and he's described a REM sleep-like state in zebrafish. So it seems like Drosophila, you had these other systems, you may have sleep and wake, then REM sleep got added in at some point and then continued, but the evidence would be it started in fish.
Mm-hmm. So you said you study sleep. Do you study like dreams as well?
No, we don't. I mean, the field started with a lot of dreams. It was very tough. I mean, would wake people up. I mean, that's where a lot of it started, right? Because Freud and the dreams were telling you a lot of information. And then what happened was people did these dream studies and wake people up and say, what are you dreaming of? Nobody could ever put a logic together around it. So people now, they're more studying things at a cellular level, what happens to this particular neuron in this particular area, that type of stuff. But dream research per se is not like it was a very important part of the field early on, but not nowadays.
So is that because people don't find it as important, or is it just because of memory console?
It's hard. I think when people did it, they couldn't really come up with a logic. They would wake people up. Then what you're dreaming about, and you try to relate to what they did in the day. Why did they dream that? Nobody could come up with a good explanation of what the dream content was. What was the dream content related to? Where did it come from? And so that's the problem.
Mm-hmm.
Now, what's interesting is there are people who get, there is a disorder in humans called REM behavior disorder. Typically occurs more common in males and females, occurs usually in your 60s. And what it is you lose this atonia. It's a so-called synucleopathy, which is a molecule that destroys neurons. And you get that in Parkinson's. So Parkinson's, and these people with REM behavior disorder, most of them over time will develop Parkinson's, right? So it's an early manifestations of Parkinson's essentially. And what it is the neurons that you're damaging in REM behavior disorder and the neurons that are driving the atonia. There's brainstem neurons that are firing out there to shut your muscles off. and they're damaged in REM behavior disorder. So what happens to people in REM behavior disorder is when they go to sleep, they live out their dreams because they're no longer paralyzed, and they're often very violent. They're often very violent. You can see the dreams, you can see videos of them. They maybe end up strangling their spouse, waking up and strangling their spouse. They may injure themselves. So the dream content is quite violent. You can actually see the dream content in these individuals because they live out their dreams.
So I know that there's this specific nucleus in the hypothalamus which regulates the sleep cycle, right? So how does that work, like on a neuron level?
Yeah, well, there is. There's a couple of key areas, but one of the key areas, as you've indicated, is the so-called preoptic area of the hypothalamus. That's where you get the galanin cells that promote sleep. And what you have is, there's other areas now, the parafacial region and the midbrain has been described recently as a sleep promoting area. But you've got a few areas in the brain that are sleep promoting, right? The neurons there fire during sleep. And then there's many areas which are wake active, right? The histamine cells in the posterior hypothalamus, the noradrenaline cells in locus coeruleus, the serotonin cells in the raphe. So there's lots of areas, the basal forebrain cholinergic cells. So there's lots of areas that are quite active and more active during wakefulness and decrease in activity during sleep. And the way it works is like a seesaw, right? When the sleep cells come on, they shut the wake cells off. They inhibit the wake cells. When the wake cells come on, they shut the sleep. So it's like a seesaw, a flip-flop. Cliff Saper at Harvard is described as a sleep-wake flip-flop. So it's interact. Unlike the clock, if you take a single cell out and you put it in a dish, you will see a circadian rhythm, right? I mean, if you got a marker in there or something changing molecularly, it's an individual cell phenomenon. Individual cells don't sleep. It's a circuit thing. It's sleep cells that are active during sleep, wake cells that are active during wakefulness, and they mutually inhibit each other. And it's like a flip-flop. That's the way it works.
So is this circadian rhythm, like this biological clock, same in everybody, or is it different?
No, the biological clock, I mean, as you know, the people got the Nobel Prize for that recently, right? For describing it. And let me explain the history of that field and then talk about the clock. So the history of the field was way back in the 1970s, Seymour Benzer, who's now deceased, he realized the power of Drosophila, fruit flies, for behavioral studies. And he was you can feed them chemicals and you can get mutagenesis. So you're knocking specific genes out, right? You don't know which genes it are, and then you're screening them to say what way they're abnormal. And they described flies who did no circadian rhythm. Some had a very short, typical periods around 24 hours, they had very short rhythm, and some had long rhythm. And that was described in the early 1970s. And then people took these flies, they kept them around, obviously. And then in the mid 80s, I think it would be, people started finding what the genes were, period gene and so on. And then the whole molecular mechanism of how the clock works was elucidated in Drosophila. It was elucidated by Mike Young, who's at Rockefeller, Jeff Hall is at Brandeis, and Mike Rojbach, who's at Brandeis. The three of them were, pretty competitive, but they worked and showed how the clock worked. And then what happened was Joe Takahashi, who's at Dallas and Texas Southwestern there, he was really doing a lot of clever stuff in mouse models. And then they identified the model, the proteins in Drosophila, they identified the homologs in mice and looked, and oh my God, everywhere had them. So the original idea was there was a clock in the brain in the suprachiasmatic nucleus. That's what kept time. Once they got the molecular mechanism, it turned out there was clocks everywhere. The lung had a clock, the heart had a clock, everybody's got a clock, right? And so the concept then came that what you have is you have a master clock. So the suprachiasmatic nucleus is like a master clock. And then it's like the conductor of the orchestra, sends signals out there to these peripheral clocks to try and get them all synchronized. So that's the way to think about it. Now, things like the master clock in the suprachiasmatic nucleus is shifted by light. So the main input to that, because typically the period of the clock is not exactly 24 hours. and it's entrained to 24 hours by the light-dark cycle. So, there's a direct pathway from the retina to the suprachiasmatic nucleus carrying light information, right? And so that's why when you go to Japan, for example, you're 12 hours out of sync, then it's a light-dark cycle that pulls your clock and gets it back in sync. But it's very slow. It only moves about an hour a day. So just an hour and a half a day. So it's very slow to move. The liver clock, the clock in the liver, it's entrained not so much by light and dark, but by food intake. And it can shift 12 hours in a day. So if you change your food pattern rapidly when you go to China or Japan, your liver clock is shifting. Your clock in the brain, he's still trying to catch up. And so that's the way it works. So there is this phenomenon of a master clock, the suprachiasmatic nucleus. It's the conductor. Light-dark information coming in is very important. And then what was realized just a few years ago, people like Russell Foster in Oxford, they made knockout mice that they knocked out a transcription factor that affect development of the eye. And the mice were blind. They had no rods and no cones. They couldn't see. But if you change the light, the circadian thing changed. So it was realized that the whole circadian system, the light information, was not as the same as vision. It was a completely separate mechanism. And then they identified it was so-called retinal ganglion cells. So there's this layer of cells in the retina, not for vision, but they send the light information to the clock. And then it was realized that these so-called retinal ganglion cells, that they're tuned to blue light. That's why you see all this blue light blocking glasses and blue light. That's why your computer shuts down, takes the blue light down at night and so on, because you're trying to take away this light information to the clock. So the light information to the clock comes from retinal ganglion cells, and they're driven by blue light, and it's not the rods and cones.
Mm-hmm. So what if these ganglion cells were damaged?
Well, yeah. If these ganglion cells were damaged, you would. And blind people, if you have damage to your retina, right, and it damages the retinal ganglion cell, you'll just free run. In other words, let's say your period was, people, they're not exactly 24 hours. Some are shorter, some are longer. Let's say your circadian period is 23 hours, right? I put you into constant darkness, and you don't get any input. And people who did these studies early on in caves and stuff, what would happen is every day you would wake up an hour earlier. You'd be moving an hour earlier every day, right? Every day you would move around. And so you can induce that with constant darkness, no other signals, no elevators, nothing to tell you what the time is, right? And you're basically so-called free run. You just do what the clock tells you. And that's what happens to people who are blind, who have damage to retinal ganglion cells. They'll free run. Now, having said that, they're being trained by the people in their house, so they'll know what the time is. So but it's challenging because they've lost the major entrainer. The major entrainer is the light-dark cycles.
Mm-hmm. I see. So are there any misconceptions in your field of study?
Well, I think the major misconception, I think, is changing dramatically. Actually, I've been in this field that I've indicated since the 1980s. In the 1980s, people would just laugh at you and say, "Doing sleep? What's your problem?" Right? And so on. And this sleep apnea, which I study, is probably one of the most common chronic disorders out there, right? And it was only originally described in 1976 clinically, although clearly there's descriptions in the literature before that of people who clearly had it. And there was an article in 1981, just not that long ago, right? 1981 by two people in Edinburgh, Scotland, saying that sleep apnea didn't exist. It was just made up by Americans trying to make money with testing people. So we had to overcome an enormous amount of bias. I never quite understood it. There's the same with sleep. People didn't take it seriously. And I think what's happened in the last few years is people are starting to really take it seriously. And I think the message, we've been at it for 30 years, 40 years, to get the message out there. And it's really happening. And then there's people I don't know if Ariana Huffington, who'd started the Huffington Post. She's a great advocate for sleep. She wrote a book called The Sleep Revolution, which is really worth reading, actually. She learned a lot about it, because what happened to her was when she was building the Huffington Post, she was divorced and she was taking her daughter, I think it was, in college visits. So she'd spend the day with her daughter. Then the daughter would go to bed. She'd work all night doing the Huffington Post stuff. She'd try to build her business. And she fell down and cracked her jaw. And she had a very, serious facial injuries from falling down. And they never were able to figure out why it happened. And they put it down to sleep deprivation. And she was just very deprived of sleep. So she became this powerful advocate. And she talks in her book about how a lot of bad decisions that CEOs have made over the years was when they were very sleep deprived, right? And I gave a talk to the Wharton people recently, maybe last year, the executive course at the business school. It was interesting. A lot of them had got it. They said, when we worked with Goldman Sachs, we were up there at night doing the trading, we thought we were great doing all these all-nighters. And now we realize it was just stupid. And they said that the guy who early in his career recognized how important sleep was Bezos, the Amazon guy. He said one of the reasons he was very successful is he never ever, he always did his sleep. So I think that there's a lot of misconceptions in the public, but I think it's rapidly changing.
Mm-hmm. Great. I see. So is there anything that you ever disagreed with as a doctor, you could say?
Dude, say that again. I disagree with people.
Yeah, any research or anything that you came across, like science?
Yeah. I mean, you get debates, and debates in science are healthy as long as they're not personal. We got a paper recently. We believe that different people with sleep apnea who have different symptom subtypes. The so-called endophenotypes, and we've documented that in a lot of studies, and then we showed in one study that it was the people who were excessively sleepy people were the ones with a cardiovascular risk. That was replicated in a study in Chile. And then a French study just came out saying they never found that. So we just wrote a letter to the journal pointing out the problems with that study. And there was many problems. But, that's healthy as long as it doesn't get personal. I mean, science, if there's healthy debate and it's all based in data, that moves it forward. If it's personal, that's not, that's destructive. So I've been involved in some of that. When I started when we started with the Drosophila stuff, all of the mammalian sleep research people thought this was crazy. They said, this is crazy. There's no sleep in these things. And yet now, when we started it, we didn't call it sleep in Drosophila. We called it a sleep-like state because we thought that was hedging our bets. Now they all just call it sleep, right? And so that debate's over. Nobody debates that it's sleep anymore. In the early days, people would rip up your papers and stuff like that. But that was based on a bias essentially. But that science corrects it. Science always corrects itself. If you're trying to do stuff like that and it's biased, I mean, science is about truth, right? I mean, there's a certain truth, and the truth's going to come out. It may take a little while and a little bumpy along the way, but in the end, the truth will well out because that's what science is.
Mm-hmm. Right. I totally agree with that.
Okay.
To sum up my last question, I just wanted to ask, who or what was your inspiration to pursue your career? And also, any general advice for younger students who want to be in a similar field to yours?
Yeah, well, I mean, let me say, I don't know how I get into this. I mean, when I was, it's interesting, I grew up, my father never went to college. He worked in a factory. He was a very smart man. The opportunity, but he was an incredibly smart guy. And I had two brothers, and my father really pushed very hard that we would get well educated, right? He saw that as if we get well educated, that would be the change. I get quite passionate about making a difference in people's lives, and that's why I ended up going to medical school. My younger brother, unfortunately now deceased, he get passionate about farming. it had nothing to do with farming. But I mean, my younger brother ended up running an enormous farm operation in Scotland with multiple 400 head of dairy cattle or something. I mean, so you find your passion. I mean, I tell people, you got to find your passion in life, because then you get up in the morning, you're keen to get going because you're doing what you like, right? And that's what you do. I think the career I've had has been fantastic. I couldn't have wished for anything better. I think science is a wonderful thing to do. It's challenging getting grants and stuff like that, and you have to be willing to work at it. But you're doing something where you're keeping your brain incredibly sharp, right? I mean, you never get dull, because up to you. I mean, it's like a one-person business. I'm going to study Drosophila. Nobody tells you to do that. You decide to do that. And if it works, you do it. And so it's an absolutely wonderful career. I know it's not as popular in the US as it used to be. People are into doing other things, but it's just a wonderful career that keeps you on top of your game, keeps you intellectually challenged your whole life. You keep reinventing yourself. And then the other aspect of science is very much an international thing, right? I mean, I've got a whole international consortium. I don't travel now, obviously, with the COVID, but I was traveling all over the place. And I've got friends all over the world. You do a lot of Zooms with them now. So you do Zooms. That's the new thing, do Zooms and so on. So you read, and it's just a lot of fun. You're making a difference, and it's a lot of fun.
I see. That's great. Thank you so much for that.
Okay.
Any last minute?
No, I think just to say thanks for doing this. I think it's great, and I think sleep and circadian research is just a wonderful area with a lot of opportunity. And nowadays, with the techniques we have available to us, we're going to make huge findings in the next few years.
All right, I see. Thank you, Dr. Pack, so much for coming. I really appreciate you and your time. I hope you have a great day.
Okay, thanks very much. Nice to meet you. Okay.
Yeah, you too.
Okay. Bye-bye.